So my name's Colin. I'm from Microsoft Research in Cambridge. We're doing this work where we're trying to treat biology as a bit of a computing problem, trying to take some ideas from computer science, but software engineering in general, and applying those ideas to biology. Now, why might we do that? A lot of the stuff that we work with is trying to produce some kind of chemicals, or we're trying to import logic into biological systems. I mean literally, like cells or larger scale systems, or like collection of those. And the reason we might want to do that is because you might be able to produce drugs that we use to treat conditions more effectively that way. But we might also be able to just directly put the logic inside people. But that's a much longer term piece of work. So the type of attack on this problem that we're making is trying to build software models to understand what's happening and also predict what's happening in other areas. I'm going to cover quite a lot of areas, and it's going to be a kind of a sampling menu of the different parts. So if people have questions as I'm going along, feel free to interrupt, and I'll try and address them. I'm going to start off with something slightly more familiar, that quite a lot of the people in the audience are programmers, but I know there's a variety of different audiences. And so we write code in these declarative, expressive forms, but eventually it all goes down to, in general, a CPU or GPU. And so this is the kind of like a view into the side of the chip. So the kind of people I know, some of them have like electron microscopes that when they get a new chip, they want to investigate what's happening. And even if you weren't involved in the production of this, you might be able to recognize structures, like notionally the different cores of execution and arithmetic units. But that's quite difficult to get a handle on. Like, there's literally so much going on, there's billions of transistors. And so what hobbyists will do is they'll attack these smaller systems. And so they'll breadboard up logic, and then they'll do this form of computation and decision making. Now, one of the interesting aspects of this is that as well as the literal form, and there's a nice form when people are discussing this, like discussing different ways they could lay out the electronics, but these two different forms, the machine over here, the implementation, and then a description of that in a kind of higher level language, which oftentimes is easier to manipulate and interact with and get a better understanding of what the intent is rather than the manifestation. Now, kind of like two of my really favorite books go into this in a lot more detail on the electronics side, and really emphasize one of the books by Petzold about the language of hardware, that there's a structure and there's an intent, and you might be able to deduce that, but you also use that to communicate to others. Now, what I'm going to try and do is do the equivalent of these books in a short lecture, but for what we're doing in biology. So to introduce you to the machines that we try and implement, and then the languages that we build to program those systems as well. So this is kind of the cell structure. What we really want to do is get inside it, identify the machines in there, and then insert additional logic. And the kind of general areas that my group works in, and I should highlight that I'm a software developer, but I work with a lot of experimentalists who work in the lab, our own, and collaborators, but also theoreticians. And so the main areas that we work in kind of go from scale of complexity, from immunology down to molecular programming. And in order to demonstrate what I want to show to people, I'm just going to focus on two areas. And so the development side of existing cells, and one feature of that, which is the self-renewal mechanism, which you may be familiar, some of you, with stem cells, and the molecular computing side of this as well. And we have some tools that we've built that help people work with that, and that's what I'm going to talk through today. So, starting with the smaller scale and building up, then this is a DNA strand, and as most of you are going to be familiar, the different components are labeled G, C, T, and A, and they themselves are these molecules that bind in this pattern, and you don't always want to think literally in terms of the machine here. Sometimes you want to have a smaller form. So down here is an exact equivalent of this labeling of the DNA helix, but unraveled just so it's easier to kind of look at. And so it's equivalent to a kind of computer code, where the computer code is binary. This has 4 different states, because each of the pairings can be one way or the other. And there are all sorts of additional rules in terms of the chemistry, but we're just going to treat it as a code that we can work with. Now, what we do with it is a bit different. So in our work, we're trying to use this as a compute fabric rather than directly interacting with the DNA genome side in this piece of work. So when you look at this picture where these bounds across, then they form like a strong connection. So it requires energy to break them apart, and they release a certain amount of energy when they're connected. The more connections there are, then the stronger the bond will be. And so in general, a long strand, when it's put into a solution, so we formulate these molecules with this code. If we put ones that are long into a solution, they'll bind tightly and they won't break apart naturally. Also, along the strands, you need kind of stronger mechanisms like enzymes, or a lot of you will be familiar with from the news, the CRISPR-Cas9 system. And so this is our basic piece of computing. And I'm going to show you how we can use that to build up logic beyond that. So the important point is the short ones, which I might refer to as toe holds, are reversible connections, and the long ones are fixed in general. But there's a case where you can unwind it, but by themselves they won't unwind. Now, it's a bit of a pain to be constantly labeling the structure, and again, this is the start of like trying to use abstraction. So what we're actually trying to do is make it unique and make it so that they don't accidentally misbind. So if we just go through and label them with a number, like 1 and 2, and the star next to the one below means it's the complement. So if you look at it before, the A and the T are in a complement form. So a 1 is going to bind onto the 1 star. Now, just to make it concrete, I just wanted to show you the kind of reactions. Yeah, good, the video's coming up. The talk comes with video entertainment as well as science. So this is a toe hold section where it makes a short connection, and next to it is this long section. Now, I said it can't just unbind itself, but what's happened here is it's been put in proximity, so it can make this random walk backwards and forwards. So this is literally, obviously there's some questions about the physics, about what really happens, but the type of thing that's happening, it's moving around, it's in proximity, so it's able to displace this longer strand. And this is the basis of the technique, DNA strand displacement. Now, again, taking the earlier section where I talked about the labeling, we have this short section which I showed in the video, which can bind reversibly, and these longer sections, which are not red for the 2. And we had this shorter section that came in and bound onto it, and then this 2 formed the displacement. So what happened at first was it had a reversible reaction connecting, and then it made this irreversible reaction underneath. Now, there are certain techniques we could break it apart with lots of heat, but under the conditions that we're running under, then this would then be stable, and this strand here would come out, and then we'd be able to attach, say, a fluorophore or a light-emitting molecule or 2 here, and then we'd be able to look in a microscope and see it. Now, we're talking about computing, and so one way to approach this problem is say we've got this digital logic, and could we implement that in this type of situation as well? And so this is let's just jump far through. And so what's happening here is a small short section's coming in from the right and pushing off this section there, and then another one's coming in from the other side. And the combination causes this to be pushed off in the middle. So remembering this whole long thing is this big long molecule along the bottom strand, and then there's these 2 short sections that are being pushed on from either side. And the combination forms basically an AND gate. Sorry, there's a question there. Pardon? Sorry, 2 and 3 different than 3 and 2? Yes, so the substructures, if you think of them as a short string, it would be like taking the second string and moving it to the front, which would mean it wouldn't necessarily bind to the same location. Yes. Okay. And so, again, I mean, even drawing out all those sections you could do on a board, but it starts to get quite irritating. And so we've developed various languages for modeling these types of systems. And so if you focus on these sections first here, this is the toe hold section, the short one, the hat symbols indicating that. Don't worry about the different types of brackets just yet, but if you have one input here, and you have another input here, and then this is the kind of backbone, which is a bit like the AND gate itself, which is what it was computing on. These numbers here are how many molecules we're going to put into solution. there's a bit of preprocessor system for instructing the system how we want to simulate it. And then I'm just going to demonstrate with one of our tools how we actually interact with this. And so flipping over to here. Yeah, and I'll make this size a little bigger. Is that fairly readable to people? Okay, good. And so we have a kind of in-software simulation of this type of system, that we have knowledge about these strands and how they behave, and so we can run a kind of computer simulation of that. And so what this is showing is input 1 and input 2, they're both being consumed, and then we're producing this other trace here, this blue trace, which is the output. So this is acting like an AND gate, but we need to test it in kind of the other variations. So if I cut down the number of one of the molecules to quite low, and also run a simulation, then we'll get some production of the output in blue, but we will not get nearly as much. And so a lot of these things are being consumed because they're making a bind onto the structure, but they're not forming a complete reaction. And so you need both in order to run this kind of reaction. So what we've got here is we've got this common section that needs to be pushed off for the reaction to happen. So there's a bit of a challenge here, and I'll give people a second to think. So this is an AND gate. How might you build an OR gate with the fundamentals? So I don't mean chaining together a lot of different gates. Does anybody have any guesses how you might instead encode with this kind of structure with displacements? Yeah, so we could build up logic and do chains of different things. But in this case, if we just try and make it out of the fundamentals, so, yeah. Yes, so that was very quick. I'm impressed. And so rather than typing it out, I'll grab it out of here. And so just as you say, there's a common section in between this 2 and of the backbone, but there's only 1 and 2 at each end. So if either of the molecules come in, that can push off the central location and carry out the computation. So it's exactly as you say. So what I've demonstrated here is kind of like the hello world of this part of the molecular programming. And it's kind of a running theme. I'm going to try and show you the very simple examples, but also give you kind of a hint at the much larger scale systems that we build. So we have a number of papers that we've worked on, and we've built whole systems of adders in analogous ways to computing circuits, but also implemented like a 4-bit square root. One of the really interesting things about these systems is that because we're throwing everything all into solution, we're not, we don't chain together everything with wires in these kind of biological systems, then everything might interact with everything else. And so if the system is assigned too much of an overlap, then you can get in situations where you'll bind to the wrong place. So we've also got a lot of analysis for trying to understand and predict where it might not produce what you intended to produce. But this is definitely a research tool, so that's just your glimpse of that, and I'll flip back to the next section.
So that problem, obviously, in biology has to occur continuously.
Yes. So what we're doing here is something very unnatural. We're taking DNA and using it as a compute fabric. Some of our other work, where we're more directly interfacing with cellular behavior, you absolutely get these like off-target effects. You get proteins that do multiple things, and that becomes really difficult for trying to infer intent. That you don't have one part of the system that does one thing, it upregulates or downregulates a lot of the other parts of it. And so you have to see these systems in aggregate and try and alter their behavior, rather than saying this thing does this one other thing and affects this one other molecule. Okay. So these are a number of like scientific publications that either have been made by our collaborators or ourselves. One of the things I found really interesting was obviously things like deep neural networks were inspired from biology and then came into computing. Somebody used this kind of system to also put a kind of neural logic back into this kind of compute. So it's a nice cycle of inspiration. There's also been a few cases where algorithms that were developed in control theory in like kind of the last century also were in evidence in biology as well. And it's kind of interesting that we might have been able to harvest some of these ideas rather than having to bring them up from nothing. Okay, so that's the kind of mechanism. I just wanted to touch on a few areas why this might be important. And so there's one of these techniques, this was referred to as DNA aptamers, that you have this problem. If your body's exposed to certain poisons or pathogens and it's completely naive to them, then it may not develop an immune response fast enough. And so what you can do is chain with some molecules between a kind of denature, a slightly altered version of a pathogen, or in this case, anthrax, and something that your system has already been immunized against. So you can imagine a small chain with chickenpox on one end and anthrax on the other end. Weirdly enough, and both of them have been made inert, so that they're not actually active. If you took that and put it into a person, the body would start to recognize that as associated, at which point the tests that were done in terms of mice is that you kind of get instant immunity. And so this is a kind of really powerful effect of being able to take the existing immunological system and then tweak it and say, this thing over here that you know is bad, it's just like that as well. And one of the other things, which is kind of more related into our work, what I find is kind of amazing, is another slightly unnatural use of DNA, is you can weave like baskets out of DNA. Then you can put little tiny locks on the front. And in the same way I was kind of talking earlier about AND gates, you can think of the two locks as if you could determine some marker for cancer or some other thing going wrong with a body, you could then more selectively target chemotherapy drugs into the locations. So all these things are very speculative, long-term proposals. You've got issues about how you get these to exist in the body. But the other big aspect, and this is one of the areas we're contributing, is that you don't want to do something like this and have accidental effects. So you really want to be sure what you're doing is what you're intending to do. And so a big part of our work is ensuring correctness. And so this is at kind of two different levels. In the same way you might test the internal consistency of a physics theory, you might also want to make sure it actually corresponds to the system that you're trying to model. So I'm going to start off talking about how we ensure our simulations are correct. And so one of the really powerful tools that we use is called FS-check. And this is I guess a lot of you are familiar with this idea of property-based testing. But I'm just going to go through a quick version to make sure it's up to speed. And so this is the kind of canonical example of how you could use quick check. If you take a list and then you reverse it twice, you expect it to be the same. And that's enabling you to test a property of the reverse mechanism on lists. And so mechanically what you do is if you get the system to generate a very large number of lists, if it finds a fault, like it might find them on edge cases, it'll then try and minimize them. So this is a way that rather than having to write hundreds of different unit tests to cover each property, you can use this higher concept of a property and teach your system how to generate it. Now, how might you generate it? And this is a very nice mechanism that's provided in functional programming type languages of using combinators. And so the FS-check library provides this ability to generate arbitrary integers. And that's fairly easy. You generate a random number and you map it onto an integer. Now, you want to build it up so it matches your own system. So again, the very simple version of this is, say you wanted to generate a pair. And so you chain together the combinators for generating integers and use that to generate lists of pairs, at which point you can do the reversal on that. So what I'm going to do is use that kind of technology, but apply it to the work I was talking earlier about, of molecular computing. And so one of the properties that we want to demonstrate is that the reactions I was talking about is that strands are conserved from the left to the right, because in those reactions, things shouldn't be snapping apart unless something else is happening. Within our model, that's not a valid operation. But we're writing a whole bunch of code, and we might make mistakes. We might join these onto the wrong places and create issues like that. So we want to ensure that our simulation is an accurate one. So what we do is we melt it, we break it apart into pieces, and we then want to test this property that taking a reaction from A to B has a property of melting A and melting B is equivalent. And so we have this reaction down here, and we want to demonstrate that they're equivalent. And so what we need to do is generate a large number of reactions. And so we have as I showed you earlier, these textual descriptions. And we also have more complicated systems that we're also making. I just want to introduce some of this syntax, but you don't need to follow along too much on the detail. And the interesting thing about this is that these are directly expressed in terms of the modeling of the system. So this is F# code, but it's fairly readable to the scientists that we work with, the systems they're trying to model. And what we can do is generate components of that in these types of structures, and then use operators to glue them all together. And that generating process, and again, you don't need to chase all the way through the text in here, but just kind of following along a bit of the detail that we generate these different components, like sections that are in the middle, sections that are in the left and the right, and little loops along the outside. We then build them up into bigger constructs. All of this stuff is being fed through the ability to generate random versions of these. So it's not just one-offs. We don't have to code in, this is the example from that paper, this is the example from that paper. Although we do that as well to ensure that we get those correct. But what we're then able to do is generate full-size gates and then feed that into the system. And we found this really interesting because we have a calculus for how these strands interact. And for the systems we tested, it was all correct, but there were certain edge cases for areas we hadn't moved into. And so we were able to use this type of approach. And one of the ways I think of this is a bit like fuzz testing, but you've guided the generation of your samples, because the system is combinatorially very complicated, and so you want to focus on certain areas, and you want to minimize it to simple areas that need fixing. Okay, and so the other side of this is very similar, that we also, for the textual description, we want to be able to pass out each of the pieces. So the little loop, we want to write how do you pass that loop from text by itself, because we might want to use it in different systems. And traditionally we used passage generators and Lex and Yak and the like, and they were great, but they were very monolithic. Whereas when we started using fparsec, we were able to write these very small combinators, and this is again a very small version of it. But what we're doing here is we're teaching it how to understand how to pass an identifier in our system, so that it's letters or digits, and we also select underscores and backslashes for certain reasons. And then this identifier here is for error handling. So we get this ability to test the parser for this by itself, but then also glue it into a larger scale system. And so we can take cases like this, and this is just a sample of our code from our full system. So just to kind of demonstrate that this is production-ready type code as well. Okay, so that's kind of described kind of the functional programming techniques that we use to kind of establish correctness of the simulations that we're making. I just want to talk a little bit how we try and ensure that what we're building corresponds to the physical world that we're trying to design and understand as well. So this is a bit of a split. So earlier when I talked about digital logic, and like one of the suggestions was we can build up a lot of these things from NAND gates, and there have been a few papers that do exactly this, demonstrating that this kind of system is the computer's showing complete, you can cascade and replicate all the digital logic in there. But it comes with quite a metabolic burden, well, an energy burden, that you have to build a lot of strands, and it's quite complicated. The interesting thing about this is that we can embed the compute directly into these interactions. And so rather than having this digital computing, we can have kind of analog style computing in a sense, where what we might want to do is say, there's a certain indicator in a cell of disease, but there's not very much of it. And how do you weigh up the reaction? Or some part of a chemical vat has a concentration, and you want to judge if it's below or above a certain level. And so what you could do is you could design a tiny little robot, and it would run around and count everything. The problem is there are trillions of molecules in there, and there's also a lot of other things going on. And so what we actually want to do is a distributed consensus detection. And so this is an algorithm implementing that. So it's a bit subtle why this works, but if you imagine a population of x's and y's, which are these strands, and when the x and the y's come together, they turn into this uncertain state. They don't know which they are. If the x bumps into somebody who's uncertain, it turns into two x's. And if the y is uncertain, and the y meets an uncertain, it turns into two y's. Now, the rate of this happening, this x, is proportional to the number of x's. So if there are already more x's, this will tend to amplify that. And equally, if there were more Ys, it'll tend to amplify this. It's quite interesting. You can actually play this as a kind of a game with people where you can pass out tokens and people will become uncertain. And at the end of the evening, it should have moved to one side or the other. But one of the nice things about this is an optimal distributed decision-making process. Though, as you may guess, there is some issue with this type of system. That what I showed you earlier was a very strict mathematical model, but we also model stochasticity. That one of the things you might have guessed is, if anybody with a background in this type of thing, is that there are only a few hundred molecules when we're simulating it. The chances are that other random noise would have occurred in the system. And so we've also got simulations for that. But what we're able to see is that on very large numbers, the probability will tend towards the consensus. And we actually have started doing this type of thing in distributed computing systems because it's too costly to have very explicit communication between all nodes. They vote and come to consensus. And so, again, another little video. This is just in case my words weren't communicating the kind of molecules meeting and bumping around and resolving. And so what we're generally moving towards was to that intermediate state with this yellow trace. And then there were more reds in the system, so they then moved off in that direction. And at which point it'll be stable, and you could then sample the solution that you had. And given that the vast majority have gone one direction, you'll then know what was in the majority before it happened. Okay. So we can take this other route rather than digital logic, that we can start encoding these different reaction types, which can then become the building blocks for building up other kinds of logic. And so this is a reversible reaction, that these short sections in the green and the red are able to bind on and push and make these random walks backwards and forwards along the molecule. And so this would just continue to move backwards and forwards. And we can also start to chain together larger sections as well. And there are certain situations where it's better to encode it into single molecules as well. But I just wanted to kind of talk through, like, these are some of the components that we can then take these high-level algorithms that we want to implement, and then build them out of those with known kind of parts, and then test in the wet lab whether they behave correctly or not. Which these are traces from one of the papers the scientists worked on, and we have the software model of what we did, and then we get fairly good traces in the solid lines. Okay. So one of the other ways of thinking about the system is that whether we want to reach certain target states. So there's a lot of work that's done in terms of satisfiability analysis in device drivers. So we don't want the device driver to reach like a buffer overflow state. And so the way to establish that is either by, again, like these exhaustive testing mechanisms, or you can do this kind of formal analysis over what's happening in the structure. And so we want to be able to ask questions like this with software programs. But there's also a kind of a direct analogy to biology. And what we need is a rich enough set of theory that we can have these traces through the systems, that we can say, like, we won't be able to hit an overflow situation because there's no pathway that can occur. And there's this analogy that can be made is the traces through a biological system. So if you imagine these chaining together of lots of reactions, we might want to be able to say that it won't exhaust the fuel when it's making choices. And so, again, starting from a very simple type of example, there's this kind of classic puzzle where you have a farmer and a goat and a cabbage and apparently his wolf. And he's on one side of the river, and I don't know if it's his pet or whether the wolf is just following him. But the boat is only big enough for pairs to travel across. And so there are certain sets of rules, which are that if the goat is left with a cabbage, the goat's going to eat it. And similarly, if the wolf is left with a goat, it's going to eat that as well. And you need to come up with a solution to that problem. It's not too hard to kind of work through it, but it's non-trivial. And so what we can do is encode in a similar kind of system to what we have. And so we can analyze. Yeah, sorry, there's multiple tabs and I have to click in the right place. We can analyze the state of the system. And then we get a nice graph. Now the problem with this is that I've encoded none of the rules. So if we zoom in a little bit, we can see that traces happen where, for example, the goat and the wolf is left alone on one side of the river, which that's not what we want at all. And so there are ways that we can add logic on top of this. So we have languages for defining predicates over these types of systems. But this is kind of a very manual, direct approach to the problem. And we've built up systems that can do this, but it doesn't scale very well. And so what we want to evoke is a similar kind of thing from computer science, a problem reduction. That this type of system has been, this decision-making system over Boolean logic has been attacked in lots of different ways, and there are very efficient, fast solvers to that. And so rather than coding up our own solutions, what we want to do is encode this type of problem into a satisfiability solver. So some of my colleagues work on that, and there's one called Z3, it's open source, it's very good. I don't directly have the sample to show the goat case for this, but there's a nice Python API and there's lots of samples around. What I wanted to talk about was, where does the complexity come where these brute force searches over the space kind of breaks down? And so this is talking about the opposite side of the problem. So before I talked about we were building up from the base up, encoding logic, but we also sometimes might want to interface with existing cells and measure what they're doing and tweak their behavior. In this case, what we want to understand is the self-renewal process. So this is a term called pluripotency, and its ability for the cells to become other cell types. So it moves into, the cell can differentiate into brain tissue or heart tissue or lung tissue, but it also reproduces the stem cells themselves. And we want to understand how that process happens. For one reason, if we could control that, we could take samples of adult tissues, apply certain chemicals to that, and then regain this stem cell-like state. And you've probably seen various news of treatments of people being treated in this manner. And there are these kind of fantastic cases where people have literally been able to see who couldn't and walk who couldn't before with treatments like this. The slightly scary thing is there's a lot of questions about what's happening here and why it's occurring. And there's also quite a strong incidence of stem cell treatment and later higher incidence of cancer. And we'd like to understand why this is and whether we can tweak that. And so we've got this model of a different system. And so what we're talking about, this is biology itself now. So these circles are genes, and a certain set of those genes are involved in this stem cell self-renewal and differentiation process. The human genome has like 20 to 25,000 genes, and they tend to act by being transcribed into proteins that act in the system and then maybe act back on the genes as well. Now, the issue is, but I'll get to that in a second. This is just to kind of show you the mechanism again. But this is a polymerase that's bound onto a piece of DNA, and what it's moving along is turning the genome into a protein by this process called transcription. This will turn into a protein that will go off and act in the system. Now, what it might do is go off and down-regulate itself. So this yellow strand that's coming off might come off and then inhibit its own reaction, or it might upregulate somewhere else in the system. But this is kind of real time as well. Okay. As in, the molecules are moving along the chain at these kind of speeds.
Is that a simulation or a visualization?
That's definitely a visualization. One of some of the videos I showed earlier, one of our problems we got was people want us to be able to visualize all our much bigger systems so you can get an idea of it. And the problem is that they took like a day to run on a compute cluster, which is a shame because that's longer than the simulation takes on our systems. On some of our bigger systems, actually, the compute time takes longer than it would take to visualize it. So maybe there's an equivalence there. So we have these genes. And they might upregulate, and what I mean by upregulate is increase the rate the other gene is acting, or it might downregulate, and we have this notation. And the problem is, if we take a mouse and we do a lot of experimental studies about where we think there are interactions between the genes. You get this giant ball of interactions. Probably the same way as if you took an automated tool and turned it onto some giant distributed system. It's like there's connections everywhere, and we don't know what's going on. And so what we want to do is understand this particular subset of the functionality. And to do that, we need to do this problem reduction. That we're taking this genes, and so I've labeled it here A, B, and C. And the black arrows are these upregulations. This red tag is this downregulation. And these dotted lines here are there might be possible interactions here. And that's what we're not sure about. So somebody might have carried out an experiment in the wet lab, and they might have done like a knockout on one of the genes, and that seems to indicate one of the other genes then stopped acting when that's happened. And so that's evidence for an interaction. The problem is it could be indirect, and so we label some of them as possible and some of them as definite that have had a more precise experiment that's been carried out over them. And so what we're doing is going to encode this, and so for people who prefer proposition logic version of it, I think some people here might, there's that as well. But what we're going to do is take this system, and we're going to take the two pathways. One, we're going to try saying, actually, the OR case is actually there. And we're then going to encode it into a very compact representation. And the other case, we're going to not have the OR present and encode it into a slightly different compact representation. And so you read it from left to right of A, B, and C being active or not. And so taken from the earlier case of the full network, these are the genes that have been were demonstrated to be part of the self-renewal and differentiation process in stem cells. So there's still quite a lot going on, and in general, it's quite easy to find evidence of there being a possible interaction, either positive or negative, but it's quite hard to tell if it's actually definitely there. And so what we did was build a tool, which again uses another computer science idea around model synthesis. So if you take a series of experiments about genes being active or not active at certain time periods, and you also take a model of that network, then you can try and synthesize possible programs that fulfill that criteria. And so in this case, the programs are graphs. And so there's another tool. I'm running this locally just so that I don't have to deal with the Wi-Fi. But this tool and all the other tools are also available on our website as well. So again, starting with the same as before, of a very simple case of that we have 4 genes here, and we have various possible interactions, and we also have potential places where we can insert observations. But I'm just going for a very simple case here of there's no observations, just try and solve this network. And this plot over here, this kind of Space Invaders plot as we term it, is the kind of summary of what's happening. So you read it from the top here, this column is a possible solution, where it's saying the A to C interaction is there and it's negative, and that these two other networks are here. And so I can show this in the graph form as well. And so this is the kind of corresponding solution to that problem. Now, of course, this is a toy model. It's just kind of for demonstrations. And as long as I haven't closed my window, this is the kind of the full network. And so this isn't going to mean a lot to most of you who don't work in genetics, but to be able to make these kind of deductions and take experimental observations and then use that to make predictions about which genes are involved and aren't involved in other interactions means you can then guide the experimentalists. So what's done at the moment, there's quite a lot of expertise and intuition about people have history of like that gene tends to be involved in that type of process. They'll go and do experiments there. But what this has allowed us to do, and with some of our collaborations with the Stem Cell Institute in Cambridge, is say, we actually think there's a lot of information content if you were to do that experiment there. And so that's led to some really powerful predictions and much more efficient use of experimental time. Okay. Jumping around. Yes, I just remembered I hadn't showed you earlier part of the kind of noise effect of these systems. So I kind of mentioned in passing that these are very smooth traces. And so this is one way to interact and kind of understand different systems. But one of the big challenges about this is that actually in low number counts, you get a lot of stochastic effects, which doesn't then correspond to biology. That it's quite surprising that like noise can be a useful mechanism. But what it allows the system to do is kind of like a bet hedging approach. And so you get this coordination between different systems. And so a more accurate simulation at these sizes is you get a lot of noise on these traces. So what I just kind of emphasize is that when we're dealing with these larger genetic systems, we get a lot of uncertainty, and that's a lot of what feeds into these. Like, there's possible interactions here, but you don't have this easy, like, turn on this switch, turn off that switch, and then you get a clear trace out of the system there. And so there's a kind of question earlier about how do you deal with systems where you get a lot of this off-target type effects, but you also have, like, this just very inherent noise. So if you're feeding one function to another function, that has noise, and then that progresses all the way through the system. Biology has evolved these mechanisms for directly utilizing that, but also compensating for it. But yeah, it's quite different work. It's what I'm demonstrating today. So yeah, one of the reasons I kept the window open is because trying to run over these very large-scale systems, even with these quite sophisticated algorithms, it's interesting. It's kind of just becoming possible. So the experimental techniques are now developing to the point where we get far more information. The software methods around satisfiability analysis and SMT solvers themselves are getting much more powerful and efficient, and the ability to express theories, and that's a big part of our work, this problem reduction, which is a huge ongoing project, which is kind of like one of the biggest reasons why I've got into functional programming so much and I appreciated it. I wouldn't dream of trying to do a lot of that stuff in a different route. But even then, we still have these very large-scale compute. So to validate our results, it's an interesting case of, it only takes about 20 minutes, but obviously I didn't want to have to run that on my laptop today. But what we're often doing is running a lot of different theories and different models and combining progressively more evidence. And so one of our researchers apparently has this enormous cluster I only just recently found out about that basically is only her work that's just churning through vast numbers of different permutations. So this kind of combination of high-level languages that are very task-specific, as well as large amounts of compute, gluing them together, lets us develop a lot more of these ideas and work with experimentalists. And so this, in a sense, is the answer to the earlier problem, that this is the minimal network where this set of genes are all that are required in order to establish the particular behaviors around self-renewal and also other parts of the essential machinery. But what we're trying to do is use this as an example of taking a piece of functionality that we can then explain in a model and then make predictions and then carry out more experiments to tie it down more deeply. And so we've had these really good results, and so that results, I mean, the best introduction to that, although it's very scientifically orientated, is a paper from a couple of years ago, and that it's a really nice application of computer science in general, of we've got ways of solving these problems, and we've got languages that we can make available to people who aren't programmers, and then we can apply these transformations and then visualize results over it. And what I really want is like more people to be inspired that these tools have become easy enough that you can kind of apply them to your own work as well. And so just kind of motivating on this side of it. I mentioned some of these before, but there's, as well as our work that's being quite fundamental and working on the design process, and we publish a lot and make this stuff available, there are actually companies that are directly taking the work around stem cells and using that to treat various diseases like diabetes. And it's this really cool application where they took cells that have been treated, put them in this semi-permeable membrane, and injected that just below the surface of people's skin. So that acts as a kind of like artificial organ almost, but in a kind of self-contained manner. And similarly, these transplants with retinal pigments into people's eyes so they could see again. And what's really nice is that a lot of these things are moving towards as I was saying, about being able to take samples from people and then treating them. Because one of the issues is that a lot of the original research around this was taking embryonic stem cells, which has a whole series of issues. But one of the biggest for the patients is that that's inherently foreign material. So although those cells can turn into other cell types, that's foreign, which your body's then going to reject. So it's a little bit analogous to having a transplant. And so these other techniques of taking samples from people, treating those cells in the correct fashion to induce these states will potentially allow people to have, in a sense, a donation from themselves, which then has none of the rejection issues. But just kind of in summary over all this, I talked about these areas around developmental biology and molecular programming, and I hope that kind of convinced that taking some of these ideas from computing of like the constructivist approach and digital logic or possibly analog logic is possible. Applying these techniques to understand existing cellular systems and the decisions and the way they work with the world is possible. I skipped over these two other sides where I talk about how some of these things actually can connect together, and that would be a different talk. But there's a whole bunch of work from colleagues of mine around tools and models and methods. A lot of these are open source. Some of them are just available in binary form. But I just want to emphasize that the work I've been talking about is there's a whole bunch of very capable scientists and other engineers who carry out a lot of the theoretical work, the experimental work, how this stuff all ties together, and then make this available. And also, as well as within Microsoft Research, we also have a number of collaborators at University of Washington here in the US and also the University of Cambridge that we take this work and we kind of move it forwards. Okay.